Okay, so two different things today. So last time we were talking about all the different kinds of receptors and pain and pleasure and touch and force and length and all different kinds of information that come into the somatosensory system. And so today I want to talk about how is that information basically used. In the case of the muscle spindles, those are the length detectors. And this, you often get this in a neuroscience class, but they kind of leave out somehow they just don't quite get to the way it really works. And so it's really about prediction. Your nervous system is making a prediction about how much something weighs. And then the muscle spindles essentially detect the deviation from that prediction. And so we'll sort of try to understand that. And then the second part is how do the pathways get into up to the thalamus and up into the cortex. And so there's a couple different pathways that we'll look at. And there's another huge pathway that goes to the cerebellum. It's actually bigger than these ones that go to the thalamus, and I'll talk about that one when we talk about the cerebellum. Okay, so first, muscle spindles, or these are stretch receptors. That's the same thing. And so for this part, I always draw a very unsightly looking arm. Okay, so here's our head of the humerus, and here's the ulna, which is your arm bone, and then we have a hand attached to it. There's our hand, and then we got a muscle. So there's the tendon where it attaches. And I'll draw it nice and big, so I have room for the diagram. So that's the muscle right there. And if you read anything about how spindles work, that thing is confusingly called the extrafusal fibers or muscle. That's the main muscle. That's the main muscle. And what moves that is what are called alpha motor neurons. Sometimes they say moto, sometimes motor, doesn't matter. Neurons. So those are the big neurons in the spinal cord that come out of the bottom of the spinal cord, and they're going that way, and they go and they synapse on the extrafusal muscle. So they make these giant end plate synapses, and that causes the muscle to spike. And when the muscle spikes, then it contracts. But then we got all the receptors, and so last time we talked about all the, Merkel, so you got like pain, temp, Merkel, Meissner, Ruffini, Pacinian. So you got all those guys, and those guys are all they're coming in through a dorsal root ganglion. I've just drawn it out in the air, but it's actually right next to the spinal cord. And so those basically are little receptors in the skin. So there's all those touch ones, and so those basically are going into the central nervous system. This is how the information gets in. But then we got the ones that are involved in the muscle, and so there was the type 1B, and those are the ones that are bringing information into the central nervous system from the tendon. So they're measuring like how much force is being applied by the muscle. But the ones we want to talk about today are the type 1A, and type 2, Roman numeral 2, muscle spindles. And so what are these guys? So these guys, they're little things that are bolted down to the muscle. So I'll draw it much bigger than, they're quite small, but I'll draw it much bigger so we can sort of see the parts. So they're actually stuck down to the muscle, and they've got their own little muscle known as the muscle spindle muscle. So they have their own little muscle, and then they got a detector part, and then another little muscle like that, and it's bolted down. So it's actually sort of attached. It's actually attached to the muscle spindle. And then what happens is, here's the information coming in. There's the ganglion cell. Here's the other end of the ganglion cell, and it goes and just wraps around that little middle detector part. And then there's another kind called type 2, and they don't have muscle spindle muscles. So they're just little detectors like that, bolted down, and there's the little squiggle of the axon. So those guys don't have muscles. So these things here are called, if you read, intrafusal, confusing, intrafusal muscle fibers. And these are the muscle spindle muscles. Muscle spindle muscles. So why do they need muscles? Kind of weird. And so there's a separate subset of neurons called gamma motor neurons, and it's unfortunate. Like, where's beta motor neurons? So you have like alpha motor neurons and gamma motor neurons, but no beta in between. So there's gamma motor neurons, and those guys synapse on the. So those are coming out of the nervous system, and they synapse right on these guys here, on the muscle spindle muscles. So that's the that's the basic setup. And so so what are the muscle spindles doing? Well, they're trying, presumably they're trying to detect the length of the muscle. But why are they detecting the length of the muscle? Well, one of the things is that there's a bunch of stuff out in the world, and it sort of weighs different amounts. You can't necessarily predict in advance exactly how much something weighs, and so how does this work? So it turns out, like I said at the beginning, you basically predict how short the muscle is going to get when you contract it. So the nervous system actually makes a prediction of it using the muscle spindles. It makes the muscle spindle muscles. It makes a prediction of how short the muscle is going to get, and then what it's set up to do is detect any deviations from that prediction. So that's fundamentally how this system works. And so, if I looked at my notes from the last year, and I had a typo in my diagram, so I'll make sure I get the diagram right this time. Okay, so if we just zoom in on this, so zoom in on this part right here, and look at the, so here's the actual attachment to the muscle. Okay, so So here we have a muscle spindle. Here's a muscle spindle. And here's the muscle spindle muscle that's attached down to the, let me make this a little bit longer. So it's attached down to the muscle itself. So this is the main muscle. Main muscle attachment. So it's bolted down to the main muscle, and this is the length of the main muscle. So we'll main muscle attach. I gotta revise my arrows. So muscle attach. So there's the main muscle attachment, and so this is the starting length of the muscle, okay? So starting length. Because when the muscle contracts, that's going to get shorter. Okay, so we're going to consider our start. So here's the starting condition. And I need. Man, my arrows are out of control here. Okay, so here's the starting length of the muscle. It's over here because I need room for another starting length. I need room for another muscle spindle example. Starting length. Okay, so now what happens when you do a contraction? So when you do a contraction, what happens is you insert a prediction as to how small the muscle is going to get. And I need one more thing, which is our dorsal root ganglion cell that's going into the central nervous system. And here's the little detector. Okay, so what you do is you make a prediction as to how much the muscle is going to contract. And how do you make that prediction? You make that prediction by contracting the muscle spindle muscles. So this whole main muscle is going to get shorter. So it starts off here, and it's going to get shorter when you contract it. So basically what the muscle spindle muscles do is they predict how much shorter it's going to get. So say it's going to get, it's going to go to here. So this is, expected amount of shortening. So say that's about here. So this is how much you expected the main muscle to contract. So that's the same thing down there. So how do the muscle spindles predict? Well, they just contract. So they're bolted down there. They just contract the amount that they expect the muscle to shrink. And if the muscle shrinks that amount when you pick something up, so here's the little detector inside the muscle spindle, then nothing happens. So you don't get any signal. So that's if you expected a load. So if I expected something to be a certain amount heavy, I sent a signal out to my muscle spindle muscles, and I contracted them at the same time as I contracted the main muscle. So the same signal, same same signal goes out to these guys. So they co-contract. The muscle spindle muscles contract at the same time that the main muscle contracts. So if everything goes right and I sent out enough of a signal to the main muscle that it correctly counteracted the load and allowed me to lift it up, then I don't get any signal. So I predicted how heavy it was going to be. I used that prediction in advance to power up my main muscle a certain amount. And so if everything goes well, no signal. So what happens if the thing is a little heavier? So this is, load heavier than expected. And this is, you don't realize how many predictions you're making. Every single time, you just do anything, like I pick up this piece of paper, I've made a prediction as to how heavy the paper is. Otherwise, when I went to pick up the paper, I would do something like, my hand would move too fast. Of course, you're all very familiar with something you didn't expect. It was a little heavier than you were expecting, and then, you pick it up, whoa, it was a little heavier than I was expecting. But that happens pretty much, just constantly. Every, thousands of times a day, you're making predictions about how heavy something is in order to determine how much to contract your main muscle. So let's examine what happens. So what happens in this case is that the muscle didn't contract as much as we were expecting. So this is, heavier load, heavier than expected. So this is, expected amount of shortening. Here's heavier than expected. So that would be down here. So now what happens? So this is the part that they usually leave out because it's so strange. So now what happens is the muscle spindle muscles will contract the same amount they did before. So these guys, so now they're bolted down here, okay? Because the main muscle has contracted, not as much as we were expecting, but it contracted. So these guys are going to contract just the same amount as they did before because, you just sent out that predictive signal. So you sent out that predictive signal here. So they contract the same amount, and so now what will happen is this guy will get stretched. And so when you look at the signal coming out of that guy, coming into the system, this guy's gonna get a stretch. So because you can see that we stretched it. Now what stretched it? That's kind of the curious thing. The muscle spindle muscles stretched it. So that's actually what caused the signal. So you strangely stretched your own receptors and made them cause a signal. So that's actually the way this works. And so this is constantly in use. Now this is a particular sort of predictive feedback thing, but separate from that, of course, you want to know where your muscles are also. So you want to know like how long your muscles are for the purpose of figuring out where your limbs are. So it's important to sort of think of how many functionally different things you're doing with these signals. So this is a particular signal about predicting like, how much the shortening should be for a given expected load. But there's other things you're going to do with this information from these muscle spindles. You also just want to know, like, where are my arms? They're flailing around. So, where is every part of my body? So that's a completely separate thing that you get from the muscle spindles. But particularly the 1A type muscle spindles are really important for this sort of predictive signal. Now, what do we do with the signal? So we didn't say what we do with the signal. So now you're going to get a signal out of these guys in this case because it was a heavier than expected load. The muscle spindles contracted just the same as they did because that's a prediction. And then that since the muscle didn't shrink as much as we were predicting, the muscle spindle muscles ended up stretching the stretch receptor, and then that went into the system. What does that do? Well, it just turns up the muscle because it was heavier than we were expecting, so just turn it up. So it's a feedback signal that just comes in. And that's pretty much, if you ever go into the, I don't know if they do this anymore, but if you go into the doctor's office back in the day, they used to have these special little rubber mallets. And what would they do? They would sort of tap on just below your kneecap. And I always, so what are you doing? So you got like, here's your leg like that. And so so there's your kneecap, here's the bottom of the leg, foot going out. So what's inside there? Well, there's a big muscle here, big thigh muscle. And what does that thigh muscle attach to? It attaches to the kneecap. So here's the kneecap. And then eventually it attaches down to this bone. So there's, you've got one bone going this way, and then you got another bone going this way. So it attaches down to the tibia there in order to sort of make your leg kick. And so what do? They come in, and you have this funny little rubber mallet like that, and you tap right on this part of the tendon right there. And so what does that do? That will stretch this guy. Now, if you think about it, that's kind of, especially if you don't know exactly when the guy's going to tap, that's an unexpected load. And so your muscle spindles won't have contracted at all because they're just sort of sitting there like nothing's going on. And so when you tap on it, what will happen? You will give a big stretch reflex to these thigh muscles, upper thigh muscles. That will stretch all these guys, and then that signal will come back into the central nervous system and say, "Whoa, that was like seriously." You seriously underestimated how heavy that was, and so then your leg will kick out. So is that a functional thing? No, that's just kind of like it's just a quick and dirty test. And a lot of these tests, like, why do you do a test like that? A lot of these tests are extremely crude. They're tests like, is your spinal cord there? Is your spinal cord basically is the circuitry of the spinal cord sort of, working, at all? Or is the whole thing kind of, messed up? So it's an extremely crude test, but that's what it's doing. It's kind of it's sort of a pathological response of this predictive system. Okay, so everybody's good on muscle spindles, how muscle spindles work. So really what's going on is the nervous system inserts a prediction through the gamma motor neurons of how much the load is, and meaning how much do we expect the muscle to contract, and that contracts these muscle spindle muscles, and if everything goes right, what happens is you get no signal. And so this basically just detects when there's been an error. Now, it can't detect on the other end, it can't detect the slack part, so it's only the positive part. It only detects like the muscle didn't contract enough. It doesn't detect the other problem, the muscle contracted too much. It mainly is like, it didn't contract enough, so we got to put more power into it and get the proper contraction. Okay, so any questions about the spindle muscles? Yeah. Oh, the same signal, yeah. Okay, so in general, it's not exactly the same signal, but in general, when people went in and recorded when they figured out like, oh, these guys are hooked up to the muscle spindle muscles, so when do they fire? And they always fire at the same time. And so why do they fire at the same time? Well, this guy is sending in the prediction as to how much shortening we're expecting, and then this guy, the alpha motor neurons, are actually shortening the main muscle. So generally, at first it was kind of confusing, like why are they always firing at the same time? Why have these two different ones? But this guy is sending in the amount of firing which causes the shortening of this little muscle spindle muscle, And that's basically the prediction as to how much the main muscle is going to shorten. And then the alpha motor neurons are actually causing the main muscle to shorten. And this is, it's just used just day in, day out. You're constantly sort of adjusting you're just like this incredible robot, kind of way better than any kind of computer robot. Just constantly, finely adjusting the input and output to do all different kinds of amazing things, like playing the piano. Okay. Is there any other questions about the spindle muscles? So it didn't say anything about the force guys. So the force guys are also sending in continuous signals about how much force the muscle has applied, but that's different than the length. So this is all just about the length of the muscle. So I didn't say anything about the type 2. So the type 2 don't have muscles, and so the type 2 ones could be a little better at just indicating just the static length of the muscle. So the type 2 might be more involved in just sort of trying to figure out, like, where is your arm based on what is the length of the arm muscles. And those guys aren't as transient, so they have a better signal. They have a more sustained signal that pretty much is more proportional to the length of the muscle. Whereas the 1A type are very transient. They just really respond when there's a change in the length of the little detector in the muscle spindle. Okay, so what we want to do is connect this up to the cortex, and so let's draw our spot here. So here's the thalamus and the midbrain, and here's the pons midbrain junction, rule of Serino. And then if we go below that, we got the fourth ventricle here. There's the fourth ventricle. Okay. So the first pathway that we're going to look at is called the dorsal column pathway. And so one question is, what is the dorsal column? So dorsal means back, so ventral means stomach, dorsal means back. So what is the dorsal column? So if we look at a spinal cord sort of in 3D, so there's the ventricle, and there's the dorsal horn, meaning sort of the back, and dorsal horn, the ventral horn, that's where the motor neurons are. And this is the white matter. I didn't label that last time, but that's all just a bunch of axons. And these are the cells. So this thing sort of, just sort of goes up to the brain. So what are the dorsal columns? So the dorsal columns are pretty much, so here's the midline. So the dorsal, there's the midline sort of heading up. So the dorsal columns are basically, it's these things here. So there's two of them. There's like a left and a right one. So, and if you actually look at the connections coming up, we mentioned last time, I'll draw it over here, but I'll just say it in words first, is that you have a dorsal root ganglion cell sitting here in the dorsal root ganglion, and that thing comes in, and when it comes in, one of the things it does is it synapses on the dorsal horn. That's how information gets up. And in some cases, like in the 1A case, it goes down and it might synapse on some motor neurons there. That's like the reflex arc that we were talking about with the muscle spindles coming in. So they not only signal the sensory part of the spinal cord, but they also go down and even touch the motor neurons. But then there's a third thing, which is what we're talking about here. And there's a branch that comes off and then heads up to the brain. So it's all coming from the same single cell. So this single cell, dorsal root ganglion cell, is sitting in the dorsal root ganglion off the spinal cord. So this is the information coming in this way. It's going to talk to the dorsal horn, maybe talk to the motor neurons if it's a 1A, and then also go all the way up to the brain. So basically this thing here is called the dorsal column. So that's why it gets its name. So now we're just kind of look at it from the top here. So now we're all the way up in the brain. So there's the fourth ventricle right there. So if you actually look at the connections from different parts of the body, you can see there's two nuclei or clusters of cells up there, and there's a smaller one, and it's kind of sitting right at. It pretty much starts where the fourth ventricle starts to open. So it's pretty much at the bottom of the pons. So this is bottom of the medulla. So this is the medulla right here, and the pons is a little bit higher up here. So pretty much just right at the base of the medulla, you have a smaller nucleus, and then alongside of it there's a bigger nucleus like that. And these have somewhat unfortunate names. So this guy is called the gracile nucleus, which means the light-bodied nucleus, the skinny nucleus. And this guy is called the cuneate nucleus, which means the nucleus that's shaped like a cuneiform. A cuneate, because it's like a wedge, sort of a wedge, like the impression in clay that you would make with the end of a reed in Mesopotamia. So that's the cuneate nucleus. And so now if we look at sort of the foot, we'll call it, this means like sort of the foot and the leg and the thigh. It's more than just the foot. We'll just call it the foot. So there's some ganglion cells there, and those guys come in. So here the ventricle is closed again, and it's closed right down through here. So that ganglion cell will come in, and it might synapse on the spinal cord, it might go to a motor neuron, who knows, but then it will go into the dorsal column like this, and it will come up and synapse on this gracile nucleus. And if we look at the hand, the greater hand, that includes the chest and part of the back. So all that goes to a visibly separate nucleus that comes up, the cuneate nucleus, like that. Now that leaves out the face. So what about the face? And that includes like part of the neck and the jaw and the scalp, a lot of other stuff. So that's going to come out. And that goes to a different nucleus. There's a nucleus that's further up here. And it has an inconvenient name, and the full name of it is there's a lot of abbreviations for it. It's the principal sensory nucleus of the trigeminal. And so what's trigeminal mean? Well, if you do gross anatomy and look at the nerve that goes to the face, it's got 3 little parts to it. It's got a little blob with 3 little parts sticking out. And so it's called trigeminal because it's got 3 parts. So sometimes, and that one is the cranial nerve 5. So sometimes it's just called, the sensory 5 or principal nucleus of 5, principal nucleus of nerve 5. The N dot stands for nerve. So it's got a lot of different names, but basically that guy is the place where the face information comes in. So it comes into a slightly different place like that, and then goes and synapses on cells in that principal sensory nucleus. And at this point, so it's sort of interesting that your brain has decided there's 2 parts of the body. It's like there's the foot part, the bottom part, and the hand part, and they're actually sort of physically separated. Of course, there's maps of all the different parts embedded in there, but even the whole nucleus is separated into these 2 chunks. And so at this point, I usually talk about spider monkeys. So if you look, so sometimes it's interesting. It's interesting when you look across different animals. You can see they have different specializations. Different animals are good at different things. Sometimes these specializations are even more visible when you look in the brain. And so an example is a spider monkey. So if you've ever been to the zoo and you see the spider monkey, I don't know if they have any spider monkeys now, but they've had them in the past. They have like a really high performance tail. And so if you look at a regular monkey, like a macaque monkey, or if you look at a cat tail, or a squirrel monkey, or a capuchin monkey, they don't have very good tails. They have like sort of low performance tail. But if you look at a spider monkey, it's basically evolved the tail into another limb. And so when you look at the spider monkey tail, if you look at the underside of it, it doesn't have any hair on it. It actually has a fingerprint. They're extremely good with their tail. They can basically just sort of like, grab onto a branch with one arm and then grab onto another branch with the tail. Or they can just hang from the tail, no problem. If you actually, a lot of times when they're eating, they'll sort of hang from the tail, hang down and sort of grab stuff. So if you look at these guys, they are extremely good with their tail, so good they can pick up a grain of rice with their tail. So they can like stick the tail out and pick up a single grain of rice with the tail. So it really is like a hand. And so if you go in the brain of a spider monkey, enterprising neurobiologists have been in there, there's a whole nother nucleus that's the same size as these guys, just for the tail. It sort of separates off, and you can see like something that's as big as the nuclei that are servicing the arms and the legs, all just for the tail. And I always say it's called the nucleus of Bischoff, and you don't have to remember that, but that's the name for it. So it's, like I said, it's interesting to look in the brain. You can see these adaptations sometimes even more clearly in the brain than when you look at the animal. Although if you look at a spider monkey, it's just like, wow, it looks like it's got another arm because it can sort of grab stuff with it. Okay, but our tail is no good. Every once in a while, somebody's born with a small tail, but it's usually taken off. We certainly can't pick up a grain of rice with it. So now let's go up into the thalamus. And so when you go up into the thalamus, so there's the third ventricle where the ventricle opens up a little bit, right, as you get up toward the forebrain. And there's a big nucleus in there, called the ventral basal nucleus. I'm drawing it bigger than it is so I can draw some parts to it. So this guy is called the ventral basal. Ventrobasal. Or VB, and it's got two parts to it. There's, the ventral posterior lateral and ventral posterior medial. But we just call it ventral basal. Now, one of the problems that comes up is, where are the different parts of it? And so it turns out, if you can just remember one thing, which is that the gracile nucleus corresponds to the foot, and it's on the midline. Or that's three things, but if you can combine those together, so a gracile, smaller, foot on the midline, then the way you can remember the maps is they reverse every time you go to the next stage. So if you go to the thalamus, what happens is we see that the face is over here. So the face is on the midline, and the foot is lateral, and the hand is in between. And so that's the opposite of what's going on here. Here we've got the foot on the midline, and then the arm, and then finally this guy is a little bit more lateral, and then we got the face. So it's kind of reversed. So here's the gracile nucleus. That guy crosses over way down in the medulla. But it doesn't hook up to anybody. It follows the rule of Serino. It goes all the way past it goes past the pons midbrain junction. This is pons midbrain junct. So it goes past there, goes all the way through the midbrain without touching anything, and then it hits the foot. And then here's the hand from the cuneate that goes and hits the hand. And then the same thing happens from the principal sensory nucleus of the trigeminal. That guy crosses over and hits the face. So if you look at the map in the thalamus, the foot is on the lateral side. If you look at the map down in the medulla, the foot is on the medial side, on the midline side. And if you ever read any neuroanatomy, this big bundle, you can actually see this bundle. It has a strange name. It's called the medial lemniscus. I've never heard that word lemniscus used for anything else than this. I'm not sure where it came from. But it's just you can just see a big bundle in the brain that's all the connections coming out of these initial nuclei that go up to the thalamus. It's important. It's kind of like the optic nerve, or one of the big pathways that's going from the geniculate up to the cortex. Okay, so finally, we get up to the cortex, and you probably know in the cortex, so here's the cortex, and here's the other side of the cortex. You probably know in the cortex that the face is lateral again. And so basically the foot is near the midline, and the hand is kind of in the middle, and the face is lateral. So like the face is down about here, and then this is about the hand here, and then this is the foot medial. And so there's yet another flip. So this guy is foot, and it goes up to the foot. This guy is hand. It sort of goes there. This guy's face, and it goes there. So it flips again. But that's our, and what is it going up to? It's going up to area 3B, and we'll talk about this next time. Area 3B and 1, somatocortex. somatosensory cortex. So that's the dorsal column pathway. It's a big pathway going up there. And so there's another pathway, a much simpler one, called the spinothalamic pathway. And to some extent, this is a functional distinction. So the spinothalamic pathway, so here's number 1. Number 2, the spinothalamic pathway has more temperature and pain in it, and light, and the sort of pleasurable sensations also. So how does the spinothalamic pathway work? So we could sort of draw it on the same diagram, or not. So let's go over here. So again, we have spinal cord coming up. We come up to the fourth ventricle, and then we got the midbrain and the thalamus here, and then we eventually go up to the cortex. So there's the cortex. Okay, so that's the pons midbrain junction right here. And there's the midbrain and the thalamus and the rest of the cortex. Okay, so spinothalamic implies that you basically are projecting from the spine to the thalamus. So it's in the name. And so what is that pathway? So that pathway, it's more pain and temperature. It's got a little touch in it, but it's mostly pain and temperature. And so let's take pain and temperature from the foot, for example. There's hand too, but here's pain and temperature from the foot. And so that comes into the spinal cord, and it synapses on cells in the spinal cord. We know that there's, like in a bunch of cells in the spinal cord, like in layer 2 of the spinal cord that have, pain and temperature signals in them. So, and then the spinal cord connects up to the thalamus. And what does it connect up to? There's the couple nuclei up there in the thalamus. These are bigger than they actually are. And there's one called the intralaminar nucleus, because it's kind of in between a couple layers. But it's a nucleus, so there's like a layer in the thalamus, in the middle of the thalamus, and then there's a bunch of cells kind of like stuck into that layer. That's the meaning of, within the layers. And then this guy's called the posterior nucleus, which is unbelievably unspecific. So it's this posterior nucleus of the thalamus. You have to put thalamus in there because there's a lot of posterior nuclei. So when you look at how that's connected up, what happens is the connections, the cells down in the spinal cord immediately cross over. But they don't break the rule of Serino because they don't connect to anybody. And so they studiously go all the way through the spinal cord, and then they go through the pons, and they go through the medulla and the pons, and then they go through the midbrain, and then they hook up to like the posterior nucleus and up to the intralaminar nuclei. And then from there, they go up to the cortex. So it's a strange setup, and this was discovered a long time ago. So back in the day, if you unfortunately got a battle axe wound that severed half of your spinal cord, which would happen sometimes, and you somehow made it through, survived the infection, you got this strange phenomenon which was detected. So here's a battle axe. Okay, so you just cut through half of your spinal cord. Okay, with an axe. So what's going to happen? Well, there's going to be some dorsal column connections on this side from the foot on this side. And those guys are going to come in, and maybe they'll synapse on some cells in the spinal cord, and then they're going to run up in the dorsal column. And they'll go up to, the ventral basal. So if we cut through the right side of the spinal cord, you can figure out what's going to happen. So what it's going to do is it's going to interrupt touch from your right foot, but it's going to interrupt pain from your left foot. And so it's a strange phenomenon where if you damage one half of the spinal cord, you've got this weird thing where like one foot cannot feel touch, but it can feel pain. The other foot, cannot feel pain, but it can feel touch. And so it's just because of this strange way in which the pathway crosses. But like I said, it doesn't synapse on anybody until it gets all the way up to the thalamus. So it basically goes through the entire spinal cord and brain before it makes a synapse. So that's the spinothalamic pathway. Now, there's another pathway. There's another pathway called the number 3, which I'm not going to talk. I'll just put it here, and that's the spinocerebellar pathway. Huge pathway. It's bigger than the dorsal column pathway, actually. People are starting to look at the cerebellum a little bit more. There's more neurons in the cerebellum than the whole rest of the brain put together. It's about like maybe 3/4 of all the neurons in your brain are in the cerebellum. So it's a big thing down there. It's small but powerful. So there's a big pathway coming up to it, which we'll talk about when we talk about the cerebellar pathway. That's the third major ascending pathway of somatosensory information. It can get to the cortex also from the cerebellum. It can get from the cerebellum into the cortex. So that's the two main ascending pathways. There's one other strange thing that I usually go over. You don't have to remember this, but if we look at this nucleus here, this principal sensory nucleus of the trigeminal. So let's draw that guy over here. So that's here's the midline. So this is principal sense nuke nerve 5. That's usually the way people write it. So that's this. It's on this major pathway, like the LGN, where you get ganglion cells come in and then they go up into the brain. But if you actually look at brain sections, what do you see? You see this giant thing hanging down from it that's like six times as big as the principal sensory nucleus. And what is that called? That's called it's called the spinal trigeminal nucleus. Spinal trigeminal nucleus. And so that one gets a bunch of somatosensory inputs. Some of the cells here go into that one as well. It's a huge, big processing station for controlling the head. So it does, all different kinds of things, chewing and all different kinds of stuff that you do with your biting, that you do with your head and face. It's heavily involved in controlling the vibrissae in a rat, because that's kind of a face thing. And like I said, if you're looking through sections of the brain, you go like, wow, where's the principal sensory nucleus? That's not very big. And then you go like, what's this thing? It's a giant thing. It's like 6 times bigger than the principal sensory nucleus. So it's a big sort of processing center for the face. And then there's another nucleus that's just slightly underneath this guy called the motor nucleus of nerve 5, because you got axons going both ways. You got axons coming in from touch on your face, but then you've got motor neurons going out to your jaws and muscles around the mouth to sort of control your body and your face. But then there's this other strange nucleus that sort of extends way up like that. And so what is that guy? So that one's called the mesencephalic nucleus of the trigeminal. And this is just a strange thing. It's in this way in all, basically, all vertebrates have this, where you see. So what is it doing? It's actually going to muscle spindles in the jaw, because biting and eating is really important. So it's going to muscle spindles in the jaw. So normally what would happen is you'd have a ganglion cell in the dorsal root ganglion, and that will go into the brain. That's sort of the normal sane thing you should do. And so what's happened with the mesencephalic nucleus is that the ganglion cells have crawled into the brain. And why did they do that? So what does it look like? So there's a bunch of ganglion cells here, and they're just hanging out in the brain. And so you can just look at a brain section, look at a rat brain section, you just find these really big cells, and they're just sort of just sitting there in a little pocket inside the brain. So they've crawled into the brain, and what they do is one end comes out, and it goes to these jaw muscle spindles, because jaw muscle spindles, it's really important to exactly control how hard you bite. If you've bitten down on something hard, your system will quickly say, like, don't bite anymore, I might crack a tooth, and then back off. And then the other side of this goes, to the principal sensory nucleus. So it's just like some weird, so we'll call that weird. So weird ganglion cells that crawled into brain. So I don't know why the brain decided to do this, but you can, you see it across all mammals have these funny cells in there. So they're not connecting up to anything in there. They're just hanging out. They just wanted to go into the brain, just hang out, have a picnic in the brain. Don't talk to anybody, in that they're next to, that the cell body is next to. So it's just one long axon basically that just comes and then bypasses the cell body and then goes down to the principal sensory nucleus. Okay, it's kind of strange. So any questions in the last -7 minutes about the ascending pathways? So we've got the dorsal column nuclei, dorsal column pathway. It's got gracile cuneate and the principal sensory nucleus of the trigeminal. And that goes across and it flips where you've got the foot on the midline, and now you got the foot laterally when you go up to ventral basal nucleus, and then from the ventral basal nucleus you go up to the cortex, area 3B, and you flip it again so that the face is lateral again. So that's the dorsal column. More touch and muscle stuff. And then you got spinothalamic pathway, where the same dorsal column nuclei synapse on spinal cells, and then the spinal cells cross over, but they don't synapse on anybody until they get all the way up to like the posterior nucleus of the thalamus. And that has more pain and temperature in it. And then we've got a whole nother, even bigger pathway that goes to the cerebellum, which we haven't talked about. Okay, everybody good on ascending pathways? Yeah. Oh, the axe? Yeah. So the axe, don't do this at home. So if you cut through just half of the spinal cord, what's going to happen is, since the dorsal column nuclei stay on the same side, so here they stay on the same, I drew the right one there, but here's the left one. They stay on the same side until they get up to the dorsal column nuclei, until they get up to the gracile and cuneate. Since they stay on the same side, this axe is going to cut, if you go through the right side of the spinal cord, it's going to cut the right foot information. But this other pathway, this other spinothalamic pathway, is coming in. It synapses on a cell in the left spinal cord, but then the left spinal cord cell crosses over instantly and then runs all the way up to the thalamus. So the crossover occurs, down here instead of like, way up here. And so what happens if you cut these two wires, you can see if I cut this wire right here on the right side, that's actually getting the left foot pain. If I cut this wire, that's getting the right foot touch. So you have this paradoxical situation where one foot can't feel touch, but it can feel pain. The other foot can't feel pain, but it can feel touch. Of course, if you cut all the way through, that's even worse. What? Yeah, so the dorsal column pathway, it has a little bit of pain in it also, it turns out. It's not, as usual, it's never all of one thing. So there's a little bit of pain in the dorsal column pathway, but it's a lot more touch and muscle stuff. So it contains touch stuff, but it also contains like the muscle spindle and the Golgi tendon organ signals in there. But there's a little pain in there too. Yeah, I did some experiments trying to map pain in the MRI magnet, and so we tried to, how can we get pure pain, no touch? Because like if you poke somebody with a needle, that's touch and pain. And so it turns out a hair removal laser is a good thing for this. So you zap somebody with a hair removal laser, and the laser heats up the skin, but it doesn't touch it. And so you just get essence of pain, just pure pain. So that way we could sort of like, we were just experimenting on ourselves. We could zap different fingers and see to look for a map of pain with the hair removal laser. So MRI compatible hair removal laser. I always joke it's a little bit of an S&M thing because you wanted like pain, but you didn't want excruciating pain. And so we had this thing where if it's not enough pain, move your hand up. If it's too much pain, pull your hand back so you can get like sort of a good moderate amount of pain in order to do the experiment. So it turns out pain is mapped just like touch is mapped. It's not surprising. Like if you pinch your little finger, wow, it hurts on the little finger. A lot of people didn't think it was mapped for some reason. It didn't make sense. Okay. So any final overtime questions about the dorsal column nuclei, spinothalamic pathway, spinocerebellar, and muscle spindles? How do muscle spindles actually work? So the idea of the brain sort of predicting something, we'll see a lot of that when we talk about eye movements. So you're kind of predicting what the world is going to look like after you make an eye movement. Here you're predicting how heavy something is going to be, so we can decide how much to lift it up. Okay. No other questions? I think we'll end the recording there, David.